Jobelyn, a multifunctional health formulation developed by Health Forever Products Limited. It is important to know whether Jobelyn has a balancing effect by not only being able to inhibit inflammatory reactions, but also be able to enhance immune defense mechanisms. Jobelyn was tested for its ability to activate several types of immune cells. The immune system is a complex system of cells involved in the defense against bacteria, viruses, and cancer. These cells interact in orchestrated ways involving activation and secretion of intercellular signals. When immune cells are activated, they send and receive signals. Activated immune cells have receptors on the cell surface to receive incoming signals and also secrete signaling compounds to other cells. Jobelyn was tested for effects at both levels. The data you will see on the following slides show that Jobelyn's immune modulating effects are not simply due to a single compound such as beta-glucan. Both the water extract and the ethanol extract showed multifaceted immune activating properties. On the graph to the left, we show the activating effect of Jobelyn on natural killer cells, and to the right, we show the activating effect of Jobelyn on killer T cells. The blue bars show the effects of water extract, and the orange bars show the effects of the ethanol extract. It is clear that the extracts have different properties. On the graph to the left, we show the activating effect of Jobelyn on T lymphocytes, and to the right, we show the activating effect of Jobelyn on monocytes. T lymphocytes and monocytes both contribute to cytokine production. Monocytes are a type of antigen-presenting cell. The ethanol-based extract has activating properties on both cell types. As I mentioned earlier, the immune cells interact with each other in orchestrated ways involving activation and secretion of intercellular signals. We have seen how Jobelyn is able to activate several types of immune cells. Now we will see data from Jobelyn's effect on secreted intercellular signals. These secreted compounds are called cytokines. Some cytokines are also called chemokines because they induce chemotaxis. We will show data on several cytokines that are involved in antiviral immune defense mechanisms. Jobelyn treatment of immune cells resulted in upregulation of numerous cytokines, both pro and anti-inflammatory. Of particular interest in light of Jobelyn's effect on killer cell activation is the specific upregulation of several chemokines involved in antiviral defenses. The three antiviral chemokines that were affected by Jobelyn are MIP-1 alpha, MIP-1 beta, and RANTES. These three chemokines were shown to suppress the viral replication of HIV in human peripheral blood monocytes. Jobelyn has several effects on human immune cells in culture that are linked to antiviral mechanism of action. Jobelyn activates NK and killer T cells. Jobelyn increases the production of antiviral chemokines. In one pilot study, the consumption of Jobelyn appeared to assist the healthier CD4 T cell count. Jobelyn has high antioxidant capacity and showed anti-inflammatory effects in multiple laboratory tests. Jobelyn treatment of human immune cells in laboratory tests resulted in increased expression of the activation marker CD69, activation of monocytes and macrophages, and changes in cytokine production. Further clinical studies are necessary to solidify the potential promise of Jobelyn as a nutraceutical support. Jobelyn has potential benefits in situations of anemia as well as chronic inflammatory conditions. Jobelyn may also offer an adjunct support in patients suffering from chronic viral infections. The goal is to advance Jobelyn from being seen as a traditional medicinal product to be accepted as a modern nutraceutical product. Science-based support of Jobelyn's effects will help gain credibility and increased acceptance of this important bioactive product.